Good afternoon. I want to bring to your attention something. There's a change in the program. Not today, but we had to juggle things around a little bit after the first posters were all made. So the posters have been corrected, but I just want to bring to your attention that next week, the topic is atopy, allergy, the common and rare allergies in the genomic era. This is really a very extraordinarily exciting business. You've probably all read something about asthma and the increasing rates and so forth. And the speakers are Josh Milner from NIAID and Pamela Guerriero, who's also from NIAID. So that's next week. Then the following week, Jeff Cohen of NIAID and Alicia Tropolnik, also from NIAID, are going to revisit shingles, herpes zoster. They actually spoke to the group almost 2 years ago, but so much has happened in that field, and the epidemic proportions have become clearer and clearer, and they've made some extraordinary advances, which I thought they should present. And the topic that had been on the 16th, the oral microbiome meets cell biology and periodontal disease, that will be presented on March 29th. So all of this is on the posters. The flood of emails will be, come out in the correct thing. But if you're looking at the old schedule, just make a note to check the website, because that's where the new schedule is, just in case there's something that you wanted to hear that you didn't. So I see we have a fair number of new faces, people here. So I'll just mention, but not belabor the point, that everybody knows that this is the Bay Bridge in San Francisco, right? This is the Brooklyn Bridge, probably the most famous bridge in the world, and you don't want to get me started about it. Why do we have this here for Demystifying Medicine? Because we are like the people on the catwalk. We are bridging the gap between advances in really exciting basic and other sciences and their relationship to major human diseases. So we're all bridge builders. So sit back and enjoy the view. There is a final exam in this course, by the way, which is electronic, and it's not based on the medical school precedent that you're supposed to memorize everything and quote back and everything else. This is ideas and concepts, and those who pass it and are here for half the sessions can receive a certificate from the NIH. Now, I want to take a couple of minutes. So this is a quiz. Old age is, this is a quote, old age is an infectious chronic disease manifested by degeneration and excessive macrophage activity, which disturbs cell equilibrium, setting up struggle with toxins, in parentheses is my addition, ending in aging and death, transforming the wild population of the intestine into a cultured population, can increase the quality of life and maybe its duration. Are there any takers as to where you think this came out of the NIH last week? You might be right. It could have, except the words would have changed a little bit. We would have read reactive oxygen and a few other things which you'll hear about today. Anybody know where this comes from? So this comes from the founder of probiotics, probably one of the greatest scientists ever to grace the planet, a man named Elie Metchnikoff, who discovered the macrophage and is well known erroneously for having pushed the idea that eating yogurt or other lactic acid producing nutrients protects the body against toxins and prolongs life. Now, there's a fascinating story about this, which I will not belabor. Second part of the quiz. Who did these experiments? When and where? Question mark. Where in the NIH would this have been done? Baby rabbits, newborn, fed only the mother's milk, are resistant to cholera. But they become susceptible when they're fed a diet that contains meat-like material. But the same rabbits, if they're fed a diet that's mainly of vegetable source, are resistant. Where does this come from? The idea that intestinal flora can produce opportunistic infections and toxins which contribute to auto-intoxication, or what we might call chronic disease. And lastly, who did the experiment where rabbits, which were given toxin-induced colitis, were protected by giving either yogurt enemas or dog feces, fecal transplant? Anybody have any idea what building this would have come out of? Well, it didn't. It all came out of Metchnikoff, who was the director of the Pasteur Institute, with Professor Roux, famous for another reason. And they won the Nobel Prize in 1908, actually for treatment of syphilis, not for this. And if you read French and want to read something really fantastic, I put this reference, and I'll put it on the website, which is the Metchnikoff view of the philosophy of life as related to the interrelationship between the intestinal bacteria and what they can do that's good as compared to what they can do that's bad, and how the rest of the body pays the price of it. And if you like it only in English, there's a wonderful article by a physician historian at the University of California in Irvine, Deborah Yan Bible, in the news of the American Society of Microbiology, and I'll put this on the web, which is the story of Metchnikoff's so-called bacillus of long life. Now, it's interesting, Metchnikoff never talked about yogurt prolonging life. And I was fascinated by this because you always associate his name with this thing, which turns out to have been a huge scandal at the time because he was Russian. This is pre-World War I. Russians were very happy in the academic environment in France. And so when he lent his name to a commercial product, La Ferme, a form of yogurt, he didn't make any money from it. He just thought it was a good idea that people should eat that. But he got blasted, and it was an Englishman who wrote a review of Metchnikoff's work and called it the bacillus of long life. He never said long life. He said better life, quality of life. And that's where we are today. We eat a sensible diet. You exercise like crazy. You live as long as you would have lived if you didn't do those things, but the quality of life, by and large, is much better. And perhaps the end is more catalytic, more catastrophic, rather than chronic disease all the way. At any rate, enough of this. So we're very fortunate indeed of having two NIH scientists of considerable renown to discuss this fascinating business of our third world, the intestinal microbiome. And you probably all know from the literature, from the popular press and everything, you go into a naturopathic store and people are selling all kinds of stuff that's going to prolong your life by changing the bugs in the intestine, the probiotic era. Well, that's the commercial aspect of it. but there's an enormous amount of science that comes first. And this is a field of explosive potential involving many, many areas. So our first speaker today is going to be Yasmine Belkaid, who, symbolically enough and quite appropriately, received her Ph.D. from the Pasteur Institute, where Metchnikoff was the director. So this is the direct lineage that's passing on to you, Yasmine. So Yasmine had a postdoctoral fellowship here in NIAID, went to Cincinnati. In 2000, she came back and was appointed a senior scientist in 2008. She's the chief of the mucosal immunology section and co-directs the entire trans NIH meta-organism initiative, the microbiomes from all over. Now, her work is particularly involved in the role of the microbiota and dietary factors in maintaining tissue immunity and homeostasis. Yasmine is going to speak first, and our second speaker, Warren Strober, is chief of the section on mucosal immunity in NIAID in the Laboratory of Host Defense. Warren graduated from the University of Rochester School of Medicine, and a year later came to the NIH in 1964, and is one of the stalwarts of this institution, primarily or greatly because his research has really been at the forefront of trying to understand this complex of inflammatory bowel diseases, Crohn's disease, ulcerative colitis, and a few others. And these two worlds intersect across the bridge. So that's what we're going to be fortunate enough to hear about. And perhaps, Yasmine, you would begin.
Thank you. Can you hear me? Yes? Okay. So thank you very much for having me here. Disclaimer, I'm just the opening act for the real star of the show. Is going to talk about IBD. What I'm going to try to do is actually discuss with you some of the main overall thematic of the field and some of the diseases that have been actually linked to IBD, to the microbiota, and not really talk about IBD per se, but I'm going to let that to Warren that can actually describe the disease. So it's interesting actually talking about Pasteur Institute, because a lot of the concepts that relate to the microbiota are really very much where something, or things that I heard very early in my thesis development, which is actually trying to understand the role of the microbiota and the word homeostasis that is actually so prevalent. In Pasteur Institute, this is a word that is used all the time. So I think a lot of my research really was inspired by people that surrounded me during my PhD. So this is just an opening slide to remind you that it's very difficult today to escape this field. Even if you don't want to talk about it, there will be probably a newspaper, a magazine in which this word is going to be presented. And the concept that the microbiota plays an important role in human health has really taken center stage recently. So I'm not going to talk too much about history. I think we had beautiful discussion earlier about Metchnikoff actually involvement and understanding of the importance of microbe in human health. But I'm just going to actually highlight a couple of points. The first one is actually that the first identification of microbes living in oral mucosa was actually done by a Dutch scientist, which name I will not pronounce because I absolutely can't, that actually really developed many different forms of microscopes. And by doing that, he was actually able to really show microbes that were actually present and actually nicely drew them with his little pencil. And actually just describe them in beautiful words that I would be surprised to see how we could send that to a journal today. But this actually was the first really identification of microbes living in high density in the oral mucosa. And I think you guys are going to hear about oral mucosa in a few weeks.
He had a very, very long life.
He did? Yeah. Yeah. Oh, yes, it's a mistake. I remember I should have. Because he actually took his probiotics. That's the message. Be a scientist and take probiotics. The second image is actually highlighting actually how you can do that by fish. That was actually a more recent image that was actually obtained in oral mucosa that highlighted actually the complexity of these microbes living in this environment. Only very few words about, again, history, because Meshnikov was actually well described, but actually we had actually earlier discovery, and the role of the important role of the microbiota as a gatekeeper was originally discovered in the context of the vaginal environment, in which lactobacilli was really linked to the prevention of infectious disease in this compartment. And that was really the first demonstration of the competitive role of the microbiota in preventing invasion by more damaging species, the concept of microbial resistance in the mucosal surfaces. And this capacity of the microbe to compete for space and resources is really the first line of defense they offer to the host by competing for space and resource. So today, it's no longer surprising, and there is actually a lot of work that has actually highlighted the importance of the microbiota in human health. And in days where you feel very good about yourself, and I think we are something, remember, you are just a minority in your own body, with actually an enormous amount of our self is in fact microbial origin. There is probably more than 10 times number of microbes than human cells, and probably more than 100 times genes expressed at any given time in the human body. So this is really what we are, an envelope that carry all these microbes that cover all the different surfaces. So the point that I actually would like to highlight is the fact that these microbes are not just present in the GI tract, which is really where most people think about these microbes. Of course, this is where the highest density of microbe is, and this is actually why IBD is actually one of the diseases that is prevalent in this site. 10 to the 14 microbes in the GI tract alone. However, these microbes are present everywhere in any surfaces. They are present also in the eye, they are present in the skin, they are present in the lung. And in all of these different compartments, these microbes really occupy unique niches and mode of interaction with the underlying tissue. For example, I'm going to come back to this point later. In the context of the skin, for example, not only the microbes are the surface of the skin, but they are also embedded in deep structures like the hair follicle sebaceous glands. And these microbes are relatively stable, although low in density. So every single surface of the human body is covered by microbiota. So importantly, we are really a meta-organism, which means a composite of microbial and human health and human cells, which means that these different components are constantly interacting and regulating each other. And there is a lot of feedback and forward loop between those different systems. The microbe influence human cells, but the human cells influence the microbiota composition and function. And this actually offers enormous possibility for actually intervention, because although our genome is actually a fixed structure, that one of our microbiota is not. So changing actually the kind of microbe we have or the function is possible. So this actually offers enormous opportunity for medical intervention by manipulating the genome or the composition of those microbes in the context of inflammatory disorders or metabolic disorders. I'm going back to this point later. So where do we acquire these microbes? These microbes that are fungi, viruses, and bacteria will be acquired at birth for the most part. There is actually some discussion, and maybe there is actually some microbe presence in the uterus environment, but it's actually not formally demonstrated. Most of the microbes really invade the different surfaces at birth. If the child is actually normally delivered, the microbiota of this child will be mostly actually associated with the vaginal microbiota. But C-section, for example, will link the microbiota of the child to the skin microbiota, and this actually has been proposed as one of the determinant events in the development of inflammatory disorders of children that are actually delivered by C-section. Then the microbiota evolve. Of course, environment is extremely important, the kind of mode of living you have, but diet in the GI tract will be a fundamental determinant of the kind of microbe that are going to be colonizing this environment. In other surfaces, what really define the composition is clear and less studied, but diet is actually an extraordinary tool by which you can actually manipulate the microbiota. Some studies have actually shown that change of the diet can actually change the microbiota function and composition in less than 24 hours. There is also change that occur in the context of development. For example, during teenager years in the skin, the entire microbiota is shifting. It's going actually from from Proteobacteria rich to basically it's changing completely because, of course, the hormonal presence in the skin and the change in the nutrient, and especially increase in actually lipids in the skin, shift completely the microbiota in this environment. It was also shown, and it's actually quite fascinating, that the last trimester of women that are pregnant and just ready to deliver, the microbiota changed dramatically, preparing potentially the microbe to be actually adequate for the child in terms of delivery. But there is really massive change in the microbiota in the last trimester of pregnancy. And of course, this microbiota keep evolving in age. I don't think it's as well studied as it should, but clearly there is a change in microbiota composition with aging. How this relates to healthy aging or not is something that remains to be actually discovered. There is actually a very big initiative worldwide to try to understand the essence of healthy aging. And these studies include, of course, the composition of the microbiota. So many different studies, and I'm not going to just describe many of them, have actually linked the microbiota to a large number of human function and physiological function. For example, of course, the microbiota is required for appropriate digestion and function of the GI tract. It is also has been linked to development of behavior, for example. In mice in which you can actually interfere with the microbiota composition, this actually could, in some cases, increase anxiety in the animal, and actually has been linked experimentally to model of autism, which of course is a model in an animal. But nonetheless, it's linked the potentiality of dysregulation of the microbiota relationship with behavior. There is, of course, an enormous amount of work to try to understand the link in the microbiota and the immune system in terms of regulating the innate and adaptive immune system. For example, the GI tract of an animal is not fully developed in absence of live microbes. And actually, you need actually the composition of this microbe to actually allow the development of the immune system structures, not just the cells themselves. So clearly, all the different functions of the physiology of the host have been linked one way or another to the microbiota. And really, there is an enormous amount of work that links almost every single physiological aspect, one way or another, to the presence of the microbes. I'm just going to give you a couple of examples in which experimental example linking the microbiota with immunity in the context of the skin, because this in fact relates to the discussion that will actually follow in the GI tract. So the skin, as I mentioned before, is also colonized by a microbiota that live in sebaceous gland or sweat glands. In this compartment, these virus, fungi, and bacteria are actually, I'm not sure this is working very well, but and you can see here an image basically of a hair follicle that was taken by a paper published by Julie Segre and Heidi Kong, where you can see the fungi that are actually lining up the hair follicle here. So as I mentioned before, they live deep embedded in the structure of the skin, and as such, the skin microbiota is important for the control of the immune system of the skin. These microbes actually are present, and it's a very interesting study that was done a few years ago at the NIH by, again, Julie Segre and Heidi Kong, highlighting how the human skin is in fact a kaleidoscope of microbial communities. And if you sample 40 different sites in a healthy individual, this will be actually 40 different communities of microbes, in this case bacteria, because the composition of the microbe is very much defined by the nutrient present in the environment. So that's actually shaped this composition. And in fact, individuals are relatively conserved because the ecological niche are relatively conserved. This is exactly the same with fungi that, again, occupy site-specific niches with a high diversity, for example, in the toenail, for example. So bacteria and fungi really are site-specific in human and relatively conserved between individuals. So this is actually how, when you want to study experimentally, you utilize animal models to study the role of the microbiota. And you need, of course, to have a system in which you can control for the kind of microbe you have. And for that, you need animals that are devoid of microbe that you can seed with differential communities. So this is actually going to be referred as germ-free mice that are really living and raised in complete absence of microbes. And these kind of animals have been used very much in many different settings, including the one where people have looked at behavior of animals. There is some interesting observation, for example, that the males actually germ-free are not aggressive compared to actually males that will be raised in the context of microbiota. So this is actually an interesting point that may be taken. So take probiotics. So this is just an example of how the microbiota control immunity. You can actually, in this case, we utilize an animal that was actually susceptible to a parasite, Leishmania major infection, and we injected them in the presence or absence of live microbes to see if the microbes that were present in the skin, in this case, were important for the control of immunity. And this actually demonstrates 2 points. The first one is the fact that if the animal has a microbiota here, they will develop a lesion due to the parasite, but also due to the immunity. In many cases, inflammation and tissue damage are caused not by the parasite, but caused by actually adaptive immunity and inflammation. As you can see here, if you infect those animals in absence of live microbe, these animals are not able to develop inflammatory responses. This is really a very small lesion that is developing. This actually really supports the idea that the microbiota is important for the control of inflammatory responses. But of course, without inflammation, you also don't have adaptive immunity. And indeed, the animals that are actually raised in absence of live microbe are incapable to develop a good immunity, here reflected by this production of interferon gamma and TNF that are important for the immune responses, that is really severely reduced when the animals do not have live microbes. This actually demonstrates that actually the microbiota can actually not only control inflammatory responses, but also is extremely important to control adaptive immunity and is in fact our primary adjuvant of the immune system. Just to show you again how you prove the association with the microbiota, because as I mentioned before, if you don't have microbe, you can have defect in development of the immune system. So you need to do an add-back experiment. And in this case, you add back one skin microbiota, it's actually Staph epidermidis, for example, and this is sufficient to restore the capacity of the animal to develop adaptive immunity against the microbe and to control the infection. So this, again, supports the idea that live microbes are actually important for the control of adaptive immunity. And this has actually been demonstrated in many different systems, both in the GI tract, in the lung, but also in the level of the skin, in which experimentally in almost every single model have been utilized, adaptive and innate immunity are really controlled directly or indirectly by the presence of the microbes. So as I mentioned before, in the normal association, there is really a positive association between the microbiota and human health. And this actually is supposed to really control positively most of the elements that we actually are required for surviving, including digestion, immune system, but also the neurological system. However, things are not always perfect. And indeed, we all know that actually diseases that affect various sites are on the rise worldwide, and in particular in places of the world in which you have actually high income level. And these diseases affect essentially tissue that are really colonized by the microbiota. And this will be the GI tract, for example. We're going to have a discussion about IBD. It will be also the lung in the context of allergic response and asthma. It could be also eczema in the context of the skin. So most of those sites that are really the home of those microbiota are also the primary target of inflammatory disease that appear to be on a dramatic rise in actually in high-income countries. And this actually is believed to be, at least in part, due to the fact that there is a dysregulation of the normal relation between actually the tissue and its microbiota. So what do we do to a microbiota? So of course, there are things that are actually intrinsic. We can really do this. It's actually our genetic hardwiring. That actually has been shown to be important to control the quality of the microbiota we have. In fact, this actually has been shown that there is strong evidence between the control of our genetic expression and actually the kind of microbe we have. There is, of course, an importance by the way you have been delivered, and that's, of course, it could be C-section or actually in the context of natural birth. The microbiota can actually be influenced also by age, and it can also be influenced by the place you live in. But there's also many other things that can be done to our environment that change the microbiota in a way that can be not very positive. For example, the way of living, exercise, for example. The diet, an aberrant diet in particular, could actually be very detrimental for the kind of microbiota that would actually seed our GI tract in particular. This actually is not just the GI tract. There is actually a study linking diet to the kind of microbiota we have in our skin, also in our lung. So clearly the diet we have influences the microbiota at large. There's, of course, the way we're actually changing hygiene. I mean, we may not have evolved to be as clean as we are. It's actually very good to live in community, but the reality is maybe that we have pushed away certain form of microbes. Overuse of actually sanitizers, overuse of antibiotics has been actually shown to be a dramatic effect on the microbiota we have in the GI tract and maybe at other compartments. And there is, of course, the presence of infection. We have actually, in the most part, evolved and co-evolved with worms, for example, that have been removed from our immune system, and that has been replaced by a different form of infection. We have a lot of acute infection in a very healthy immune system. That means that this system is constantly challenged by acute challenges. And this may actually change the kind of microbe we have, maybe not for the best. So there is actually a lot of work recently trying to understand the kind of causative association between disruption of those microbiota composition and the development of inflammatory disorders. And as such, a large number of inflammatory disorders have been linked to dysregulation of the microbiota composition. And this will be, of course, IBD, that is believed to be due to a collapse of a relationship with the microbiota. But also in the context of cancer, there is actually experimental evidence, and I'm going to come back to that later, that linked different kind of microbes with the development of cancer, and actually has been shown also to interfere or to promote cancer treatment. There is actually, of course, a link in the microbiota and anxiety, as I mentioned before, but in fact, most of the disorders that have been studied so far, including autoimmune disorders, but also inflammatory disorders, have been linked one way or another to the microbiota. So again, a field of enormous interest and growing development. At the moment, a large number of the studies remain experimental, but there is growing actually clinical evidence linking the microbiota to different disorders, and I'm going to come back to that later. So I'm going to give you a few examples in the literature, and especially examples that have actually linked not only experimental model, but also clinical evidence. So some of the first work that linked really the microbiota with disease recently was the work that was done by Jeff Gordon that really had seminal observation linking the kind of microbe you have and obesity. And what actually was done over the years was actually to transfer microbiota from actually obese host to actually lean host and show actually that just the transfer was sufficient to transfer gain weight. And I'm describing here actually an experiment that is actually an extremely well-controlled experiment because they use discordant twins. And those discordant twins were discordant for weight, and they took the microbiota of an obese twin and a lean twin, transferred them in a germ-free animal, as I mentioned before, and this was absolutely sufficient to transfer gain weight. It's a remarkable observation that shows, and in fact, in some individuals, aberrant growth of certain microbes may actually predispose to obesity, something that, of course, can actually predispose to metabolic syndromes.
The obese twin was made obese
by life. So basically, they had different kind of life, and this is not that they were made obese in the context of the study, but they were actually basically one was obese and one was not, and they were the causation of the obesity is unclear, but they were actually obese. One was them, so they were actually naturally obese. So another thing that actually has been proposed recently is the overuse of antibiotics as a dramatic set point for the predisposition of inflammatory disorders, and in context also of obesity. So a similar experiment, in this case that was done by Marty Blazer, that actually showed that if you actually give low-dose antibiotics in mice, you can actually, in fact, predispose those mice to obesity again. And again, this was actually, now they actually did clinical observation showing that actually use of antibiotic early in life in children may also predispose to gain weight. So this is actually a fascinating observation, and in fact has been used for a long time by farmers that utilize this strategy to make their cows gain more weight. In fact, they give low amount of antibiotics as a mean to make sure those animals gain weight. So this actually is an important observation because clearly antibiotics have been on the dramatic overused in West high-income countries, not always for bacterial infection, and this clearly is something that can predispose to severe long-term consequences. So there is actually a lot of, yes? So in the case, so in the question is some antibiotics are more prone to induce these kind of things than others. Which one? Yes. Yeah, so it's a very interesting question. So in the study that I mentioned, I don't think this was the case. I think they use actually low-dose broad-spectrum antibiotics, but I suspect this is actually has been done. And actually, I'm sure that actually some class of antibiotics may be more predisposing than others, and I'm sure this is actually in the literature. So another form of disease that actually has been linked to the microbiota and really received a lot of attention recently is diabetes, both type 1 and type 2 diabetes, actually. The first evidence linking type 1 diabetes and microbiota was done experimentally in mice, in which using actually a model mice that were susceptible to type 1 diabetes, they found that actually changing the microbiota was actually an element that could actually predispose or not to the induction of the disease. But more recently, actually, there is some fascinating observation that links actually in human the microbiota composition with actually type 2 diabetes development, which is another extremely important finding. And a very interesting observation, for example, that was done actually recently that showed that usage of sweeteners in food may actually be very detrimental because it allows the outgrowth of microbes that themselves are sufficient to predispose to glucose intolerance. So there is really a fascinating body of work that really linked the microbiota with the development of many inflammatory disorders, and diabetes is actually one of them. A lot of work is also done trying to link the microbiota and cancer development. In fact, again, experimentally, the first work was done by actually using, in fact, it was not experimentally, it was actually human studies that show, for example, that Fusobacterium was highly enriched in colon cancer of patients. And experimentally, those bacteria were taken and actually put back in animals that are predisposed to cancer development, and showed again that this transfer of these bacteria was sufficient to transfer, to promote actually susceptibility to cancer development. Again, a growing body of work trying to understand the association between those microbes and the development of cancer. But many of the things that those microbes do are actually causative and potentially causative. They can induce DNA damage, they can alter beta-catenin signaling, and it can, of course, increase pro-inflammatory signals. So again, a lot of research is currently done trying to understand the association between different microbes and cancer development. Work that was done in part at the NIH, but also in other places of the world, really highlighted the importance of defined microbe in the GI tract in the context of immunotherapy and therapy and chemotherapy. This actually studies show that if you change the microbiota of an animal, you can actually promote or prevent the capacity of the host to respond to treatment. And this finding is quite fascinating because it was now actually reproduced in many different settings. Not only those two treatments, but also PD-1 treatment on anti-CTLA-4 have been shown actually to also being associated with the kind of microbiota you have. And in a fascinating study that was published recently, it was shown that if you take the microbiota of people that failed CTLA-4, anti-CTLA-4 blockade treatment for therapy and transfer it to animals, you can actually transfer the failure of treatment. So again, a fascinating observation that showed that how maybe very easily we can actually promote treatment or the efficiency of treatment of cancer. So I'm going to finish by really some of the spectacular observation and work that was done in C. difficile infection. And I'm going to discuss that because this actually is probably going to be a proof of concept that may change the field. So C. difficile is a very important disease that affects a very high number of people, especially in clinical settings. It is still the cause of a large number of deaths in America, but worldwide, especially in people that will be immunosuppressed or in hospital settings. And really, the only kind of treatment that really seems to have worked over the last years is, in fact, a fecal transplant. And remarkably, proof of concept that was actually published a few years ago demonstrated that really over 95% of people can actually be cured just by a fecal transplant. Again, if we had actually observed farmer before, we would have known that this was actually known. And actually, farmers themselves have used for generation and generation fecal transplant as a mean of treating cows that were infected. In fact, that's actually how they treat a severely chronic bacterial infection. But this is actually quite remarkable, and it's actually led to fascinating discovery in the field. This is actually some of the work done by Eric Paymer. And what he did in this study was actually to now try to just, instead of doing fecal transplant, that of course had some issues because transferring the microbiota from one person to another could also create metabolic syndrome. It actually was done in human, unfortunately. Could also potentially transfer pathogen that we are not aware of. So this is not clearly something that will be a viable approach for the long term. So what Eric Paymer did was to try to understand and develop algorithms to try to understand what would be the best predators of C. difficile in the context of an ecological setting of the GI tract, and really define a microbiota that would be a predictive of control of this infection and devise a strategy in animals to actually control the infection. So it's a really rational approach of this treatment by trying to understand what are the best predator of this pathogen. So why this is actually fascinating is because this is going to become probably one of the most developed treatment for C. difficile. But this actually led to the development of drugs. And there is actually the first drug that is now approved by the FDA is actually a drug that is actually going to treat C. difficile. And the way by which this was actually approved by the FDA is because the company, that I'm not going to show the name of because I'm at the NIH, utilize spores, which is basically something that you can quantify and you can actually make in a way that is actually compatible with pharmaceutical development. And this actually is going to change the field because until then, people were very skeptical. How are we going to just utilize the microbiota for treatment? And this actually is the proof that you can actually develop a drug based on the microbiota. And this is actually now on phase 3 of clinical trial, but actually they're also trying that in the context of IBD. And actually this is something that's going to probably just now bring the microbiota in the clinic. So I'm just going to this is actually my last slide. That is actually my introduction to Warren Strober. Just to say that IBD has been linked to a collapse of a relationship with the microbiota. It's a complex disease. And really, this microbiota presence has been believed to be one of the important initiator or amplifier of this disorder. And actually, we're going to have now a beautiful discussion on this topic. Maybe you want to take a few questions before? Yeah? No?
Please speak about
You early on you mentioned the difference between vaginal birth and C-section.
Excellent.
How long does that difference in the microbiome last in life? Is it
No, it's not
years
permanent.
long?
No, it's not permanent.
Not permanent.
It's not permanent, but actually this is actually, the first few months, the few years of life going to be determinant. So this change is not permanent because then, of course, going to be remodeled by environmental exposure and diet, etc. But clearly, there is an early shift, and if you actually look at neonates for the first few months, they're actually different from them.
Is there anything known about the mechanism of, say, antibiotic-induced obesity? What happens to those calories?
What happens to those calories?
Well, you might reduce competition for calories, but I don't know whether that's a significant addition to the problem.
So the mechanism per se remains a bit nebulous at this stage. So I think clearly there is the kind of microbes themselves are less efficient, and they're less efficient to actually metabolize the food in a way that is appropriate. That's one of the elements that actually contribute to the aberrant accumulation of calories. The other thing is could be also indirect. The microbiota also themselves can actually regulate absorption. They can also create metabolites that are important in the context. So there is actually, I think, the way by which the microbiota interfere with appropriate accumulation of calories is probably at many different levels, not only controlling the function of the epithelial cells, but also themselves not being capable to metabolize appropriately the food that is actually provided.
Well, can you isolate anything from the bacteria which are removed by the antibiotic?
Themselves as a product. Yeah. So no, I don't think it has been shown that you can actually take a single product from the microbiota and actually transfer gain weight or not. I think it's going to be more as an ecological response. But inflammation per se can actually promote obesity. So that's another element that the microbiota can indirectly contribute to obesity by increasing inflammation. So it could really act at many different levels.
Can you speak to the preliminary association between the microbiome and neurological conditions such as autism?
Yeah, I mean, of course, I will just remain extremely cautious. This is experimental data, and I think this is a very sensitive subject that should be approached with a lot of caution. Experimentally, I can explain to you what they have actually done. What was actually done in the context of this study was to take animals that if you provide, I think it was poly-IC during pregnancy, can actually develop some of the defect, social defect that actually has been, so for example, the mice are less social, they're more anxious. And in these animals, if you actually have certain kind of microbes at birth or not, or if the mother has certain kind of microbe before delivery, you can prevent or mitigate some of the effect that is actually developed. So these studies suggest that actually the microbiota limit inflammatory responses that become detrimental to the development of the fetus. Now, in the context of adult animals, it's different. It was actually shown that you can transfer different kind of microbiota and transfer anxiety. So it seems to suggest that actually anxiety itself can actually be promoted by certain microbes. Mechanistically, those things remain a little bit nebulous, but I think this is actually where the field is. We are at the stage of actually collecting remarkable observation, and I think it's going to take years to understand mechanism.
So Yasmine, how close do you think we are to routine microbiome testing, where people profile their microbiota to evaluate whether it's healthy or not? And do you think there's an understanding for what constitutes a healthy microbiota?
No.
Is it dependent on where you live in the world, what your ethnic background?
No, you're absolutely correct. I don't think there is a consensus of what is a healthy microbiome. And I think when I use the word dysbiosis, it's a word to take with caution, because there will not be a fingerprint of the appropriate microbiota. Your microbiome may be healthy for you and may be detrimental for me. I think we are far from actually trying to understand what is the ideal microbiota for each person. But I think in terms of actually knowing the kind of microbe we have, this is something that is going to come to the clinic very quickly. I think this is becoming simpler and simpler. And I will imagine at one point, relatively soon, when everyone coming at the clinical center will actually be evaluated for the kind of microbiota they have and how the treatment they had changed the kind of microbiota. And this could be important because it was also shown that how you metabolize a drug is also controlled by the kind of microbe you have. So efficiency of treatment could also be impaired or accelerated by some kind of microbe you have. So,
Hi. I noticed that throughout your talk you mentioned live microbes. Have you transferred non-live microbes and constituted the same kind of phenotype in these mice?
yes. So in the context of how the microbes are really doing that, so there will be different kind of elements, but so far, at least for us, we found that most of the effect is due to live microbes. It's been relatively challenging to find, single molecules. So there is actually a couple of molecules, for example, PSA, that is produced by Bacteroides, can actually have and reproduce some of the effect of the bacteria itself. But in the vast majority, live microbes seems to be important for the reproduction of the overall effect. They live in the unique niche. They have a very specific mode of interaction. And I think what we need to understand now is how we can actually leverage this kind of understanding and deliver it as a drug. So that's.
Yasmine, I noticed on the slide where you showed the multiple systems that are affected by the microbiome, you had behavior.
Yeah.
Can you explain that?
Behavior was more actually about the anxiety behavior or fear. And actually, there is some fascinating studies when you look at people that do behavioral studies in mice, showing really that you can change the behavior of animals by changing the microbiota. And this actually was also done in the context of this study of autism models. So, yeah. So if you're in bad mood, just say it's your microbiota. That's just going to.
So let me slip in one other question. We're going to get to this with the periodontal disease.
Yeah.
But do the formation of biofilm, what is the role of the microbiome in the production of a biofilm?
Well, it is the biofilm.
It is.
Yeah. So the microbiota.
So the intestine really has.
Actually, there are biofilms almost everywhere.
Biofilm.
Absolutely. So biofilm, as clinicians, people think of biofilm as like, ooh, this is actually going to be an infection system. In fact, a lot of microbes can live as biofilm. The ones that are living in our mouth are biofilms, and they are the microbiota. So in the GI tract, it was shown that some microbes live under normal circumstances as biofilms. So there is actually healthy biofilm formation that are really important. There is also a lot of communication between bacteria. Quorum sensing is actually a very important mechanism of interaction, also in the context of the microbiome itself. So those microbes are organized and they communicate with each other. Yeah.
I thought the biofilm had something to do with resistance to antimicrobials.
In the context of pathogenic infection, absolutely, it's going to prevent. Actually, it's going to just maintain the pathogen and resist to treatment. But actually, microbes that are healthy microbes can actually organize themselves as a biofilm under normal circumstances. Yeah.
Okay, well, there'll be time. Oh, no, there won't, because Yasmine has to leave at 5:30. But thank you very, very much. That was it. Thank you. Thanks again. I'll come and visit.
Well, it's a pleasure to be here again. I've given variations of this lecture for a number of years, and each year we have to change because this is such a rapidly developing field. I want to thank Yasmine for this really extraordinary beginning, and it makes my lecture much more simple to deliver. Well, inflammatory bowel disease is really two different diseases that are really quite different. One is Crohn's disease and the other is ulcerative colitis. I'm not going to talk about ulcerative colitis very much today because of time considerations, but I will talk about Crohn's disease quite a bit. And what is Crohn's disease like? Well, it's a chronic, relapsing, full-thickness inflammation of the gut. It affects the small bowel, affects the large bowel, the colon, or both in about equal numbers of cases. It can cause fistula, which are connections between the gut and the skin or other structures. It could cause abdominal pain, bleeding, diarrhea, weight loss, fever, and lethargy. It's associated with a number of extraintestinal manifestations listed here. And the patients can be about split between 70% chronic relapsing and 70% of patients require some sort of surgery. You can see here the area of narrowing of the gut that occurs. You can see some of the skin manifestations, and you can see over here the granulomatous inflammation that occurs in these patients. It can be a very, very difficult disease indeed. Now, over the years, we've developed a concept of the disease which has been quite useful, and that is that the disease is caused by a dysregulated response to mucosal ligands and antigens. So you can see that on your left, the normal gut is in homeostasis with the bacterial microbiome, or indeed fungal, viral microbiome as well. And there's relatively little inflammation, and there's a normal control between effector cell responses and regulatory cell responses. But on your right is the inflamed gut, and somehow the same ligands and bacteria now cause inflammation in the cell populations in the gut, the dendritic cells, macrophages, and the T cells. There's the elaboration of effector cytokines such as interferon gamma and IL-17 and TNF alpha. And in fact, many of the biologic means of controlling these diseases depends on giving anti-TNF. It's very effective in a large percentage of patients. Coming on board is the use of anti-IL-12 P40, which is directed against the IL-17 response and the interferon response. So these are very effective mechanisms of treatment. I'm going to start by talking in general terms about the microbiome and gut inflammation, and then give you some highly developed biochemical studies to show you how one can dig down and understand how the microbiome can actually affect the cells of the gut and cause inflammation. Now, there's been a great deal of study of the nature of the microbiome abnormalities, both in normal individuals and patients with Crohn's disease, other patients with ulcerative colitis. This is more or less a summary of what has been found at the phyllo level. The firmicutes are depleted, and the proteobacteria are increased in subsets of patients. The abnormalities tend to be limited to the small bowel and are more prevalent in Crohn's disease patients than in ulcerative colitis. Bacteroidetes are decreased or increased depending on the study, and a particular organism, which is a member of the Clostridium family called Faecalibacterium prausnitzii, is decreased. I'm going to talk more about that in a few minutes. There's no evidence that there is a pathogenic organism in the gut of these patients. So these are the normal bacterial species, and they're reacting to the in the gut environments to cause inflammation because of pathologic reasons. Now, there seems to be a division of labor in the gut between various organisms and the kinds of responses they can elicit. There's a good deal of study about Bacteroides fragilis, for instance, which has a bacterial coat called PSA, or polysaccharide surface antigen, PSA. This has been shown by Yasmine and others to induce cells to produce TGF-beta 2, and then to induce FOXP3 regulatory cells, FOXP3 positive regulatory cells. So this is considered to be a major type of bacterium that can induce a regulatory cell response. And this would be protective. This would prevent disease. And whether this does so in the normal individual is still under investigation. Now, in the middle is a very important group of bacteria, the Clostridium. Clostridium species. We've already heard about Clostridium difficile causing infection. I'm going to talk about this to some extent. This induces cells, epithelial cells, to produce TGF-beta, which in turn induces FOXP3 regulatory cells. So here again, there is a protective response, prevention of infection of inflammation. On your right is the SFB. These are organisms that have been newly discovered by Dan Littman and his associates. They adhere to the epithelium. They induce cells in a complex way. Serum amyloid A is induced, which then induces dendritic cells to produce IL-17, become IL-17 cells. This is an important way in which IL-17 cells, effector cells, are induced in the gut. Now, the organisms in the gut microbiome, as I've already implied, either can prevent or cause intestinal inflammation. The ones that can induce regulatory responses, and I'm going to talk considerably about Clostridium organisms, induce regulatory T cells. On the other hand, certain kinds of E. coli members are found in the gut of Crohn's patients in particular. They called adherent inductive E. coli, AIEC, can enter macrophages and induce macrophages to produce effector cytokines. We'll talk about that as well. Now let's talk for a moment about the clostridial organisms. This has been extensively studied by a number of Japanese workers. Honda, in particular, has led a group of workers. They have found that certain members of the Clostridium family, clusters 4, 14A, and also 18, not shown here, can induce cells, dendritic cells, to induce irregulatory T cells. Exactly how the clostridial organisms actually induce these regulatory cells is still under investigation, but it's thought that they elaborate short chain fatty acids, butyrates in particular, which then act on epithelial cells. And the epithelial cells then induce substances to act on dendritic cells to make them more inductive of regulatory responses. So this is a very important finding, but whether it applies to humans or not is still not known. It certainly applies to mice. And if you take germ-free mice, for instance, and get populations of clostridial organisms and inject them into germ-free mice, they will manifest a much higher level of regulatory T cells and be protected from various inflammatory conditions. Now, you can talk about, we just talked about regulatory bacteria that induce regulatory cells. One could also talk about cholinergic organisms that induce inflammation. And I want to make a distinction here, a very important distinction. Cholitogenic organisms can be defined as organisms that, if introduced into a normal host, will induce inflammation in the normal host. There doesn't have to be an inflammation or an abnormality present in that host. That would be a true cholitogenic organism. There's been only one situation in which a true cholitogenic organism has been found. It was studies of so-called truck mice, complex mice. They're RAG2 deficient. They lack T cells. They're also deficient in a transcription factor known as TBET. They make a lot of TNF alpha. They make a lot of IL-17 and IL-23. And they get a colitis. The remarkable thing about these mice is that if you take the gut microbiota from these mice and put them into normal hosts, they will cause disease in normal hosts. The recipient animal could be completely normal. So if you co-house normal animals with the truck mice, the normal animals will get colitis, at least for a certain period of time. These are then true cholitogenic organisms because they can cause disease in normal hosts. Now, in contrast to that, are cholitogenic organisms that develop in NLRP3 deficient mice. These are animals that lack a particular kind of inflammasome that is present specifically in the epithelial cells. These animals develop colitis due to particular organisms. One of them is Prevotella organisms. But they'll only cause disease when you also give the animal DSS colitis. You have to treat the animal with a substance, a chemical, dextran sulfate, which has a negative effect on the epithelium, causes the epithelium to become much more permeable to organisms. So these are cholitogenic organisms, but they only cause disease in an already diseased host. So they're not completely cholitogenic. They require the host to have an abnormality. Now, how does this manifest itself? If you look at NOD2 deficient mice, now we're going to talk a little bit more about NOD2 deficiency later on. NOD2 is a gene that encodes a protein which react to muramyl dipeptide, part of the protein encode of bacteria. And you can give these mice this DSS colitis. That is, you can treat them with dextran sulfide and induce a colitis. And remarkably, what you see here, the DSS colitis animals lose weight. And this is a study of either the single-house mice or the co-housed wild-type mice. You can see the co-housed wild-type mice lose more weight because they're exposed to the flora of the NOD2 deficient mice. And this, just direct your attention to the colored bars. You can see that the NOD2 deficient animal has a lot more bacteroidetes and a lot less firmicutes, shown in blue. So the firmicutes are the subtype of bacteria, the phyla, that has the Clostridium. So in having less firmicutes, you can assume that they have less clostridial organisms and therefore less organisms that are capable of inducing regulatory cells. And that may be one of the reasons why they develop more DSS colitis. Now, I talked about the microbiota that induce FOXP3 regulatory cells, and therefore they're protective. And I also talked about, to some extent, the E. coli, the adherent penetrating E. coli. These are organisms that occur in patients with Crohn's disease, not all patients, but some patients with Crohn's disease. They get into macrophages and they induce the macrophages to produce TNF. So these adherent invasive E. coli, or AIEC, are a colitis producing or inducing organism, but they're probably not causing the disease. They are members of the cholitogenic subpopulation that can cause disease when there is already disease in the host, such as Crohn's disease. So this organism can be found in normal individuals and doesn't cause a disease in normal individuals. It can be caused in high numbers of cancer patients. It doesn't cause colitis in the cancer patients. So there's many reasons to believe that they're secondary. They are occurring within an inflammatory environment. They bind to the epithelium through a protein called CCAM6. They enter the lamina propria through ulcers that are generated through the inflammation. They can aggravate disease, but they can't cause disease primarily. And in fact, they're antibiotic, they can be killed by antibiotics, but patients with Crohn's disease are not really susceptible to antibiotic treatment. If they were, then these E. coli would be controlled by the antibiotics and the patients would get better. We're not advancing. We have the we're not advancing. Yeah. Oh, here we are. We're going. Now we're okay. Yeah. Now I'd like to shift gears a little bit, having talked a little bit of general about cholitogenic organisms, types of cholitogenic organisms, also organisms that induce regulatory cells, and organisms that induce effector cells that cause inflammation. And I'd like to turn our attention to NOD2. NOD2 is a gene of which there are certain polymorphisms, and certain polymorphisms occurring in the NOD2 gene have been associated with Crohn's disease. This, in fact, is the first genetic association that has been found to be associated with Crohn's disease, and is the most common genetic association. It is coded by card 15, and it is an intracellular, intracytoplasmic sensor of muramyl dipeptide, a component of peptidoglycan. About 10 to 15% of patients with Crohn's disease bear CARD15 polymorphisms or mutations. Well, now the NOD2 gene consists of a CARD region, a NBT, a NOD region, and a leucine repeat region. The mutations generally occur in the leucine repeat regions, and that's where the molecule recognizes the MDP. And as a result of that, you get Crohn's disease. Now, what is the reason why NOD2 mutations cause Crohn's disease? It has to do with the way NOD2 interacts with the microbiome. The first theory that was put forward is that since NOD2 also occurs in epithelial cells and so-called Paneth cells, which are kinds of epithelial cell, you need the NOD2 to produce certain antibacterial substances called defensins. And as a result of lacking NOD2, there's less defensin production, there's an explosion of bacteria in the gut, and this causes the inflammation. The problem with that theory is that, in fact, the best data available shows that any abnormality of defensin production that occurs is actually secondary to the inflammation. It's not a primary abnormality. And it's not really well proven that NOD2 acts through reducing defensin production in humans. Now, another theory, which is a theory that we developed here a number of years ago, is that NOD2 is very important in allowing cells to respond with regulatory, in a regulatory fashion. Namely, if you stimulate the animal with NOD2, with MDP, through NOD2, you get greatly reduced immune responses. So in this complicated slide, I just wanted to direct your attention is to compare the pink and the yellow. These are responses to various TLR ligands, peptidoglycan, double-stranded RNA, LPS, flagellum, and so on and so on. If you compare the pink and the yellow, the pink is the primary response. The yellow is if you stimulated the cells first with MDP and then came back and gave it a second stimulation. In the second stimulation, the response is much lower. So, you can either compare the upper line, which is IL-12P40 responses, or the lower, which is the IL-6 responses. No matter which TLR ligand you see, if you compare the pink and the yellow, the pre-stimulation of the cells with MDP gives you a much lower response. Now, we looked at this very carefully as to what the biochemical mechanism was, and we looked at various proteins that could be produced as a result of MDP stimulation. And we found that MDP stimulation induces a lot of a substance called IRF4, shown circled in red. IRF4 is a very important protein in the cell. It's involved in B cell development. It's been shown recently to be very involved in differentiation of dendritic cells. So it turns out that MDP can induce a lot of IRF4, and you can assume that perhaps the lack of IRF4 production in the polymorphic, in the animals with, or the humans with the mutations, can lead to trouble. Now, we did a very simple experiment. We had an animal model of inflammation induced by a substance called trinitrobenzene sulfonic acid, which is given intrarectally. When this is given intrarectally, shown in the blue line, the animals lose weight and develop inflammation in the gut. However, if at the same time you give the animal muramyl dipeptide, this is the substance that induces NOD2 responses. If you give the animal muramyl dipeptide, you can completely prevent the development of the inflammation. It's a very striking phenomenon. So here's a substance that's normally present in the gut because it's part of the bacterial coat of both gram-positive and gram-negative organisms. But if you give an excess amount of this material, you can prevent inflammation. And this striking result has been repeated in various laboratories around the world. Now, you can also show that this happens in another model, which I've already mentioned, DSS colitis. And shown here is that animals lose weight, but if you give muramyl dipeptide, shown in the orange, they don't lose weight. They're protected from the colitis in DSS colitis. But if they lack NOD2, because they're NOD2 knockouts, and that's shown in the green line, even if they're given MDP, now if they lack NOD2, they are not protected by the MDP. However, if you direct your attention to the right side, if you have an animal that lacks IRF4, we were fortunate at this time to be able to get these knockout animals. Now the MDP does not protect the animal. The animal has to have IRF4. So this increase in IRF4 that MDP induces is somehow very important in protecting the animal from the development of colitis as a result of seeing MDP. So at this point, we postulated the following idea, that MDP can get into the cell and induce or stimulate NOD2. The NOD2 then somehow interacts with IRF4. The IRF4 somehow blocks the development of the NF-kappa B responses that are ultimately responsible for the inflammation, the IL-12, the interferon gamma, and the IL-17. So how is that really happening? What is going on? How is the MDP acting through IRF4 to prevent inflammation? And I'll stop here to say that this is the interaction of the microbiome with the host immune system, because the microbiome contains these, this MDP that can influence the development of the NOD2 response. Now, shown here is that when you stimulate either NOD1 or NOD2, NOD1 is a related protein, you stimulate NOD1 or NOD2, one of the things that happens is that a downstream kinase called RIP2 or RIC, has two names, RIP2 or RIC. The RIP2 is ubiquitinated, and it has a certain kind of ubiquitination. It's called K63 ubiquitination, polyubiquitination. This is associated with activation of the molecules. The RIP2 becomes activated when it undergoes K63 polyubiquitination. It has become it's slated for degradation when it undergoes K48 ubiquitination. The ubiquitinated RIP2 then interacts with downstream molecules, one of the most important of which is TAC1, or another one is TRAF6, and then this initiates the NF-kappa B cascade. So we looked at this very, very carefully. If you remember what I said, is that what we found is that if you pre-stimulated cells with muranyl dipeptide, you got a blunted response, you got a decreased response, you got a tolerogenic response. If you look here, you can see that if you pre-stimulate cells with MDP, that's shown as the first stimulation. What happens, if you look down, is that the IRF4 is induced, as we saw before. But in addition to that, IRF4 then interacts with RIC, which is a downstream molecule. It interacts with MYD88 and then interacts with TRAF6. So the pre-stimulation with MDP is inducing RIC to interact with IRF4 and TRAF6. And this is also shown in a very, very nice way with dual link staining. I'm not going to go into this because of time. But you can also show that NOD2 pre-stimulation induces IRF4 binding to RIC, TRAF6, and NYD88. Now let's go on to polyubiquitination. You can see here that if you pre-stimulate cells with MDP, ubiquitination is shown as a smear. How do I get back now? Okay. When you pre-stimulate with muranyl dipeptide, you get a smear, which is the molecule that has been ubiquitinated. And what you could see is that pre-stimulation induces, in this case, the RIC to undergo K63. What the pre-stimulation do is prevent the polyubiquitination, the K63 ubiquitination of RIC. Also prevents the K63 polyubiquitination of TRAF6. But it doesn't have any effect on the K48 polyubiquitination. And you can also show that if you reduce the amount of RF4, we're using an siRNA to RF4, you can prevent this prevention of polyubiquitination. So the effect of the MDP is to prevent the polyubiquitination of downstream signaling molecules. Now, if we go back to what we saw before, you administer MDP to animals with TMS colitis, you again see that the addition of MDP prevents the colitis. The animals don't lose weight, and they don't activate the NF-kappa B system. So they're protected from colitis. But they also, the MDP treatment also induces IRF4 interactions and inhibition of the K63 polyubiquitation within the colon. And what this slide shows is that as a result of MDP treatment, there's lack of IR4 interaction with RIC, that's shown with red. You get interaction of IR4 treatment with RIC. You get IR4 interaction with MYD88 and with TRAF6 as a result of the MDP treatment. And we can go one step further, and we can actually administer an IR4 expressing plasmid to the animal. So you actually administer the plasmid, and the plasmid does the same thing as the MDP itself. It prevents the inflammation. So as a result of giving the plasmid, the animal manifests reduced amount of activation of MDP and interaction with RIC, MYD88, and TRAF6. So in the end, what we see here is that as a result of muramyl dipeptide, you get activation of NOD2, induction of RIC, which then interacts with IRF4. The IRF4 then prevents the polyubiquitination of various downstream molecules, such as TRAF6 and MYD88, and that prevents the activation of NF-kappa B. So in this very complex way, we see that the gut microbiome, through its interaction with NOD2, is downregulating the immune response. And in patients who lack NOD2 activity because of a genetic defect, they don't have this regulation, and you get an increased response and you get inflammation, which we call Crohn's disease, at least in the experimental animal. And in fact, other investigators have shown that in actual patients with Crohn's disease, you get decreased NOD2 responses. So this is the sort of, if you dig down and look at the biochemistry, the way the gut microbiome can interact and regulate the immune system. And if that regulation is not occurring, then you get inflammation. Thank you.
Very insightful. We had some questions. Well, people are thinking, so are there therapeutic aspects involved with muramyl dipeptide?
Well, one thing that we've been thinking about is administering MDP to patients. And we're collaborating with people at Johns Hopkins to create nanoparticles that embed MDP. You can actually give it to patients and induce regulatory responses, but only in those patients who don't have the polymorphism and can't respond to MDP.
Yeah.
So that in the 95% of patients with Crohn's disease who can respond to NOD2, this would be effective therapy.
So why do Crohn's disease patients get granulomas and not overt non-granulomatous inflammation?
That's a good question, and I think it relates to the fact that you get a lot of activation of macrophages in this disease, whereas in ulcerative colitis, which is non-granulomatous, it's more of a superficial inflammation affecting the epithelial cells. You don't get that much activation of macrophages.
Does this, you would imply that the same mechanism is active in ulcerative colitis?
Oh, yes. And ulcerative colitis is a very different kind of mechanism. But I would imply that what's happening here is the activation of macrophages and dendritic cells.
It's not too polymorphism.
Wait a minute. Just a minute. Go ahead.
So it looks like or generally people use just weight just to track if the colitis is happening or not, or inflammatory bowel disease. Are there any kind of biochemical markers for that?
For the animals or
Yeah.
the humans?
Let's start with animals.
Well, there's no specific biochemical markers. You get an inflammation, but the inflammation is rather nonspecific.
Is the presence of the polymorphisms associated with Crohn's disease, is that sufficient? In other words, that most people
No, it isn't sufficient because, in fact, most patients who have the polymorphism don't have the disease. So this is the case with most polymorphisms. There are over 150 polymorphisms that have been associated with inflammatory bowel disease, 150 to 200. In almost every case, a polymorphism itself cannot cause the disease. It has to be combined with other abnormalities.
So what do we know about the patient who has an acute episode and then goes into a prolonged remission for years and years? Can you actually mechanistically conceive of what's going on there?
Well, it could be that, for instance, there's a development of regulatory cells or IL-10.
Yeah.
So that the response is actually can be sort of polymorphic in a sense. You can also induce regulatory cells. And we've shown that in certain strains, under certain conditions, the non-2 deficiency could actually be protective because at certain stages of the disease, you actually have more regulatory cells.
I see. Are there any other questions? Well, if not, thank you very much. That was a tour de force. Wonderful.